Dynamic state of body constituents concept of metabolism. Okay, one of the greatest discoveries ever made was the observation that all these biomolecules have a turnover. That means they're constantly being changed into some other biomolecules or also made from some other biomolecules. They're changed into some, made from some, and all. It's always in this transit. And this sum total of all the chemical reactions, that is your anabolism and catabolism, that is your metabolism. Okay, and I guess it's okay. So for example, removal of CO2 from amino acid, making an amino acid into an amine, removal of amino group in a nucleus, all of these, hydrolysis of a glycosidic bond in a disaccharide. See, when you have a disaccharide, let's say you have lactose, you have glucose and galactose, they are connected with your glycosidic bond. So when you're hydrolyzing, that is adding water to it, the bond is going to break because they are bonded by dehydration. But when you add water, when you're hydrolyzing them, the bond is broken. So that's that's a metabolic reaction. So we can list many of those. Majority of these metabolic reactions do not occur in isolation, and they are always linked to some other reactions. In other words, metabolites are converted into each other in a series of linked reactions called metabolic pathways. So this let's say glycolysis, you have 10 steps. So first you have your glucose, that's converted into pyruvate is converted into other compounds. So it's always this compound from this is again in this is again reacting to form with this. So it's always one metabolite converting into other. It's all are, it's never an isolated reaction. It's always in a series or linked reactions. Okay, that is called metabolic pathway. And every chemical reaction in our human body is a catalyzed reaction. Okay, this one, when you're performing some reaction, you can make it normally without using any catalyst, or you can increase the rate of the reaction by adding a catalyst. In our body, every chemical reaction is catalyzed, and the catalyst form that is used here are your enzymes. And enzymes, except ribozyme, which is a nucleic acid, all are proteins. Okay, so there's no uncatalyzed metabolic conversion in living organisms. In human body, I just meant living organisms. Okay, even CO2 dissolving in water, a physical process, or even your hydrolysis, that is also a catalytic process in living organisms. So the catalyst hasten the reaction rate of given metabolic conversion are also proteins. The catalyst, they are your enzymes, except ribozyme, all are proteins. Okay, that's what I've written. Metabolic pathways, a more complex structure, can lead to, let's say, anabolic, can be from simpler structure you're forming a complex structure, or let's say catabolic, from a complex structure you're breaking it down to simpler structures. So basically that building up is anabolic, breaking down is catabolic. Anabolic, you're building it up. So basically you are giving that energy, you are forming bonds. You are forming bonds between that molecules, so you have to give it energy. So it's an endothermic reaction. You are giving energy there. Energy is consumed. In catabolic reactions, let's say even your oxidation reaction, in human body, when glucose is broken down, aerobic respiration, ATP is formed. What is ATP? Energy. Energy is released. Energy is released when there's breakdown of glucose into your CO2 and H2O in aerobic and lactic acid and anaerobic respiration. So catabolic pathways lead to release of energy, anabolic lead to consumption of energy. Glycolysis, this metabolic pathway from glucose to lactic acid, which occurs in 10 metabolic steps, is called glycolysis. I'll say something. Living organisms have learned to trap this energy. Okay, the most important form of energy currency in living systems is your ATP, that is adenosine triphosphate. And okay, the living state. Basically, living state is always a steady state to achieve equilibrium. Like, we are never in equilibrium. Okay, equilibrium means death. That's it. When you're at equilibrium, all your reactions are just stopped. They come to a standstill. Okay, so the living state is a non-equilibrium steady state to be able to perform work. When you're at equilibrium, you can't perform any work. This is one of the thermodynamics law, second law of thermodynamics. Okay, so that's what it is. So just remember this data. The blood concentration of blood glucose level in normal healthy individuals is 4.2 millimoles per liter to 6.1 millimoles per liter, and your hormones are even more less, like nanograms per ml. So this studies any okay, as living organisms we work continuously, so we cannot afford to achieve equilibrium. So, and this non-equilibrium, this is achieved by energy input always. So metabolism provides a mechanism for production of energy, and hence the living state and metabolism are synonymous. And without metabolism, we know no living organism can survive. It's not a living state basically. Okay, enzymes are almost all of them are proteins except ribozyme, which is a nucleic acid. nucleic acid that behave like enzymes. So an enzyme, like any protein, has a primary structure, that is amino acid sequence. It also has secondary and tertiary structure. The backbone of the protein chain folds upon itself, the chain crisscrosses itself, and hence many crevices or pockets are made. Just look at this tertiary structure of protein. You find these hollow places, right? These are your active sites. This is the enzyme. This is enzyme. Enzyme. In this enzyme, you have some hollow sites, hollow places that are active sites. These are the sites where the substrate attaches to the enzyme, and this, when substrate attaches to the enzyme, it forms a product. Okay? These are called crevices or pockets, as you see here. One such pocket is the active site. An active site of an enzyme is a crevice or pocket into which the substrate fits. Thus, enzymes through their active site catalyze reactions at a high rate. So, the enzymes catalyze, basically, they are catalysts, as we saw earlier, as I said earlier. So, when substrate, so it's basically like the substrate is there, it forms, let's say, product, something, in the presence of this enzyme. Okay? Like the substrate attaches to the enzyme and that forms the product. That's the overview of basically how it works. And we also know that there are many inorganic catalysts. How are these organic catalysts different from inorganic ones? One main, there are many others, but one main is that inorganic catalysts work efficiently at high temperatures and high pressures, while enzymes get damaged at high temperatures, say above 40 degrees. Like we human beings, when temperatures are different, like we have enzymes have an optimum temperature at which they work. When that is not there, let's say you're increasing the temperature at high temperatures or low temperatures, they don't function well. Enzymes can't, because these proteins either degenerate or they break down or they just become non-functional, temporarily inactive. Okay, so that's why inorganic, they work at high temps and high pressures, whereas organic do not work. However, some enzymes, but we know that archaebacteria, they are there in high temperatures, high acidity, very high pressure, and all those. So those enzymes, their enzymes are attuned to survive in those high temperatures, though their enzymes work in those. So thermal stability is thus an important quality of such enzymes, isolated from thermophilic organisms. So thermal stability is what you find in thermophilic organisms. That's one thing. Thermophilic, basically, they love living in high temperatures. Chemical reactions, there are two kinds. One is your physical reactions, chemical reactions, like physical processes, chemical processes. In physical processes, you basically have either change in shape, change in state of matter, let's say water to ice to water, water to vapor. That's a physical process. Any process where you do not have any bond formation or breakdown, without any change in bonds, that is a physical process. Okay, it's always like external, without breaking any bonds. Whereas in chemical, you have breaking of bonds or you have formation of bonds. Let's say hydrolysis of starch. When you're hydrolyzing starch, which is a polymer of alpha glucose, you see that glucose are being broken down. Okay, the starch is broken down into its constituents, that is glucose. So that's a bond breaking. So that's why it's a chemical reaction. And this is an inorganic chemical reaction. As you can see, of course, it's a displacement, bonds are being broken and formed and all those. Similarly, hydrolysis, as I told, of starch, which forms glucose, is an organic chemical reaction. And the rate of physical or chemical process refers to the amount of product formed per unit time. Okay. It can be expressed as rate is del P by del T, the amount of product formed by time, of course. And this rate can also be called as velocity, and if direction is specified. And rates of physical and chemical processes are influenced by temperature. Okay, this general rule of thumb is that when the rate doubles or decreases by half for every 10 degrees change in either direction. So when you're increasing 10 degrees Celsius, the rate doubles. Okay, when you're decreasing by 10 degrees, the rate halves. Okay, that's what it means. Catalyzed reactions proceed at rates very high than uncatalyzed. Okay, catalyzed ones, you have a catalyst. Of course, the rate of reaction is going to be vastly higher than the uncatalyzed ones. For example, in the absence, see this, carbon dioxide plus water forms carbonic acid. Okay, basic. Without the enzyme carbonic anhydrase, you see that 200 molecules of H2CO3. 200 molecules of H2CO3 in the absence of any enzyme formed in 1 hour. 200 molecules of H2CO3 formed in 1 hour. Whereas in the presence, by using the enzyme carbonic anhydrase, which is present in cytoplasm, the reaction speeds dramatically, about 600,000 molecules being formed every second. Literally. So the enzyme has accelerated the reaction by about 10 million times. That's the power of enzymes. So there are thousands of enzymes catalyzing different metabolic reactions, and this is not just single step, it's a multi-step chemical reaction where different enzymes are used and different enzyme complexes are formed, and that all constitutes a metabolic pathway. Okay, in glycolysis, again, that's mentioned, 10 different Okay, sorry, not glycolysis. Okay. For example, this is actually a metabolic pathway in which glycose becomes pyruvic acid through 10 different enzymes catalyze different metabolic reactions. 10 different enzyme catalyze metabolic reactions. And then, okay, in our skeletal muscle, under anaerobic conditions, lactic acid is formed. Under aerobic conditions, pyruvic acid is formed. And in yeast, the fermentation, same pathway, leads to the production of ethanol. So different conditions lead to different products formation. So you understand anaerobic lactic acid, aerobic pyruvic acid. In yeast, by fermentation, you have ethyl alcohol formation. So how do enzymes bring about such high rates of chemical conversion? This is because, enzymes basically, I showed here. Okay, enzymes, they are this structure where they have this sites, that is your active site, where the substrate binds and this forms into product. So here again, the chemical which is converted into product is called the substrate. It has active sites, hence enzymes, that is proteins with 3-dimensional structures including an active site, convert a substrate into a product, symbolically S to P. So it's now understood the substrate has to bind to the enzyme at this active site within a given cleft or pocket or crevice that we learned. The substrate has to diffuse towards the active site, and thus an obligatory formation of ES complex, that is enzyme substrate complex, is formed. This complex is in a transient state always, like during the state where substrate is bound to the enzyme active site, a new structure of the substrate called transition state structure is formed. Okay, basically this is what I'm going to explain here. Okay, the same paragraph I'll explain with this. You have your substrate plus enzyme, which forms an ES complex, which is short-lived. It's always in transient state. It's an unstable structure. It can be a single structure or it can be multiple structures, and these all intermediate structures are unstable. They are unstable. Okay, they're all short-lived, they're all unstable, and then they form EP complex, that is enzyme product complex. This is divided, this is broken down further into product and your enzyme, and this enzyme, this 3-degree structure protein, is unchanged and can be reused again in the reaction. Okay, that is what is explained in this. Okay, now we'll learn about stability. So in to know stability, I'll just explain this thing. So stability is achieved when the potential energy of something is the least. So let's say there's a molecule Na. Okay, I'll explain with a graph. Okay, you say this is your graph. Sorry, just saying this. This is your potential energy. This is your compounds, let's say, whatever. Okay, let's say Na is there. Na, let's say it has this much. Its potential energy is this. And let's say Cl also has this much. But when NaCl is formed, it will have the least potential energy. Okay, why? Because potential energy, or basically energy, is inversely proportional to stability. Or let's write stability. Okay, this is the whole point. Let's say you have a ball that's kept here. Okay, and you have a ball that's kept here. Okay, if you give a slight push, it's going to fall. But here if you give that equally slight push, it's going to come back to its state. So this, which has more H, right, this has the negative actually H. So its potential energy is in negative. Okay, negative potential energy is the least potential energy. So hence this ball is more stable and this is least stable, or least stable. Okay, that's what is the whole point here. Potential energy mgh, let's say same mass, same gravitation, but the thing is this has high potential energy, so it's less stable. This has low potential energy, so hence more stable. That's the whole point. And here you see, now this graph I'll explain. Substrate is there, it has some potential energy for itself, and as the reaction progresses, let's say the product has the least potential energy. It's actually the energy is decreasing, the product's energy is less than that of the substrate. So don't see the intermediate, just focus on the starting and end points. We see that the energy of product is less than the energy of substrate, so hence this energy is released. The energy here is released, hence this is an exothermic reaction. If suppose you have a graph, say like this, here I've done this. Let's say substrate's energy, this is potential energy, this is progress of reaction. Substrate is here, and this, just forget about this, this is where the product is ending. Okay, product's energy is more than the substrate's. This is because this metabolic reaction required energy, so you are providing energy, so energy is used, energy is consumed. This is an endothermic reaction. Okay, whether it's exothermic or endothermic, here you see now, here you have always okay, this line is very important. Implicit intermediate structures. In the statement, the fact that all other intermediate structural state, basically what the meaning is that all the intermediate structural states are unstable. So as you're progressing in the reaction, these intermediates are being formed. So somewhere like this, your intermediates are being formed, your stability is reducing, so potential energy is increasing. So like this, and you get this maximum where the last unstable structure is formed, let's say. Okay, and then slowly we are reaching stability. Slowly EP complex is formed. Slowly here they are dissociating and forming the product. So always without the presence of enzyme, you are having a activation energy, the most activation energy. Here with enzyme, the least activation energy. So activation energy is basically the energy difference between the average energy of substrate and your highest potential energy of the intermediate compound, like at the transition state, whatever you're having the highest. So that's what we are understanding from this. So as we progress through the reaction, you need some energy, you need activation energy always, even in endothermic, even in exothermic. But ultimately the product would be in exothermic least potential energy, in endothermic higher potential energy. That's the whole point. You can ask that this is not stable, this will undergo many other reactions, I guess. Like you're having let's say your body's muscle buildup, it's forming muscle, but it's also broken down when you're not, let's say when you didn't eat for a long time, so muscle mass is broken down or your fat areas are broken down. So yeah, it's always in a transit. I think this is what I explained just now. The difference in average energy content of a substrate from that of its transition state is called activation energy. That's what I explained. Yeah. Now this one I explained just now actually. Your E plus S enzyme to substrate forms ES complex, which forms EP complex, that is enzyme product complex, which forms your enzyme and the product. The most important one is the catalytic cycle in the next page. This was also PYQ, and this they asked like arrange this in the right order of steps. Okay, so first the substrate binds to the active site of the enzyme, fitting into the active site, and then the binding of the substrate, because the substrate are binded to the enzyme, this induces the enzyme to alter its shape. Like it makes the enzyme is like trying to change its shape for the substrate to bind better. So go a little fit more tightly around the substrate. This active site of the enzyme, now in close proximity to the substrate, breaks the chemical bonds of the substrate, and now enzyme product complex is formed. The enzyme releases the products of the reaction, and this free enzyme is ready to bind to another molecule of the substrate and run through the catalytic cycle once again. This is what the catalytic cycle entails, I guess. Yeah. Then you have factors affecting enzyme activity. So the factors basically are your temperature, pH, concentration of substrate, and presence of an inhibitor or something, presence of different chemicals in the like wherever the reaction is taking place. Okay, so yeah, temperature and pH, you see this graph. I'm explaining this here with the help of graph. Always pH and temperature have a maxima, like they work the best, they work optimally, efficiently in this optimal pH and optimal temperature, this optimum temperature and optimum pH. Whenever you are reducing that pH or increasing that pH, reducing the temperature or increasing, their rates are going to go down. Okay, they work the best maximum enzyme activity in their optimal range, and it's going to decrease as you go further from them. And then you have this this point, low temperature preserves the enzymes in a temporarily inactive state. Like low temperature keeps it in an inactive state, the enzymes go, and in high temperature it destroys the enzymatic activity because proteins are denatured by heat. That's it. This one is again important, concentration of substrate. Just remember, when you're mentioning concentration, it's written like the substrate ka concentration, let's say A ka concentration, like this brackets, square brackets indicate concentration. So substrate concentration and velocity of reaction. Now let's say you have an enzyme which has 10 active sites. So you gave it, you're keeping on adding substrates. Okay, as you're adding, the rate of reaction is increasing, but when all the 10 are filled, you see slowly the rate is decreasing. As initially it's increasing, but slowly it reduces and it reaches a maximum where all the 10 sites are binded and you cannot bind anymore. That is your maximum velocity. Now Micheli's constant is the concentration of substrate at which half of maximum velocity is achieved. To see, Micheli's constant is Km, like constant K, M Micheli. It's the concentration of substrate at which maximum velocity by 2 is achieved. Half of maximum velocity is achieved. Affinity between substrate and enzyme is inversely proportional to, just look at this, inversely proportional to Micheli's constant. So if Micheli's Okay, so when when substrate and enzyme are very fast binding, so the rate of reaction is very fast, so that we maximum velocity by 2 is achieved very soon, like very fast. If the rate of reaction is lower, like let's say like this, and that Vmax by 2 is still here, you see the Michaelis constant would be somewhere here. If Michaelis constant is more, the rate of reaction is less, which means affinity between substrate and enzyme is low. Hence, affinity of substrate and enzyme is inversely proportional to Michaelis constant. That's what this means. And the next para tells exactly the same. Increase in substrate concentration. With that increase, the velocity of enzymatic reaction rises first. Ultimately, it reaches a maximum velocity which is not exceeded by any further rise in concentration of substrate. This is because the enzyme molecules are fewer than the substrate molecules. After saturation of these molecules, there are no free enzyme molecules to bind with the additional substrate molecules. Okay, for this, like your enzyme molecules are getting lower than your substrate molecules, and after a point, there are no free enzyme active sites for the substrate to bind. That's the whole point. And now inhibitor. Okay, just listen to this. Okay, when you have an enzyme with 10 active sites. Again, enzyme with 10 active sites. You have a substrate A, there are 10 molecules of substrate A. So in normal conditions, this substrate A, 10 molecules of substrate A bind with the 10 active sites, and you get 10 products, let's say. But let's say there's another 10 molecules of some B chemical compound, something which is structurally very similar to the substrate A. Your B is similar to A. So let's say, for simplicity's sake, now this B is competing with the active sites with A. So now, for simplicity, we are saying that 5 substrate molecules are binding and 5 B molecules are binding, which results in only 5 products to be formed. Whereas in the first case, in normal conditions, you're getting 10 products. So the B compound is inhibiting the formation of product that can be formed by the A substrate. So that is the work of competitive inhibitor. The activity of an enzyme is also sensitive to the presence of specific chemicals that bind to the enzyme. When the binding of the chemical shuts off enzymatic activity, the process is called inhibition. Like if the B is binding to the enzyme's active site, it shuts off the production of, it shuts off actually what the enzymatic activity should do. So it's called inhibition, and the chemical is called inhibitor. When the inhibitor closely resembles the substrate in its molecular structure and inhibits the activity of enzyme, okay, it is known as competitive inhibitor. So the inhibitor competes with the substrate. Okay, due to its close structural similarity with, I'll just say this again. Okay, close structural similarity with the substrate, the inhibitor competes with the substrate for the substrate's binding site of the enzyme. Consequently, the substrate cannot bind. As a result, the enzyme action declines. Now we have an example: inhibition of succinic dehydrogenase by malonate, which closely resembles the substrate's succinate in structure. Such competitive inhibitors often used. Okay, I'll explain both. You have your succinic. Okay, you have your, let's say, succinate, which is the substrate, and some enzyme is there, which in that presence you form succinic dehydrogenase. Okay, but when malonate is present, which is very structurally similar with succinate, it inhibits the production or formation of succinic dehydrogenase. That's what they mean. The inhibition of succinic dehydrogenase by malonate, which closely resembles the substrate succinate in structure. Such competitive inhibitors are often used in the control of bacterial pathogens. Okay, you have your antibiotics. When bacterial infection happens, what your bacteria does is that it changes the little wiring of your cells. Like your cells usually form normal proteins when you're healthy, but when bacterial pathogen enters, it forms a different kind of protein which the bacteria feeds on, thus growing. So what the antibiotics do is that when you're taking antibiotics, they have a chemical that, let's say the bacteria is forming some substrate that binds to some enzyme. This substrate, they make sure that this antibiotic is similar in structure with the substrate and is more reactive, of course, and connects to that enzyme, thus preventing the bacteria substrate to bind, inhibiting bacterial substrate. Thus bacterial proteins are not formed. So thus bacteria die. They can't feed, they are not growing, they die. That's the whole mechanism of antibiotics. And then there's, okay, classification and nomenclature of enzymes.